So, and this is a course that is open to the TRI program as well as some people in Methodist. But so we'll walk you through this today. This is the first lecture, so welcome everyone in the class. And we do start out actually with a bit of a survey, and to give you a bit of background of where we're coming from. Each of the instructors and team will give you a little bit of background. This was not made by me, but thanks, Taylor made a draft of this, and I feel a bit sheepish putting a big picture of my face up there, but it's obviously me. And so, I've also written a bit about long-term plans for going down the planets. Actually, if I was writing an email that said, Dr. Mason, I have a plan to make moss that could grow on Mars. So writing books like this leads to interesting emails like that, which are interesting to think about for an iGEM class, which we'll talk about in some of the later lectures, but I'm a big enthusiast for technology and its applications to medicine, of course, here at the medical school, but also long-term application that could help us quite literally survive someday on other planets. Which is becoming more possible because of other technologies that we won't talk as much about in this course, but of course from SpaceX, Sierra Space and Blue Origin making a lot of exciting new developments in that sector. But to tell you a bit about my background, we've been looking at single molecule sequencing. If you, for example, looking at Google Scholar or PubMed, we've been thinking about this actually for really decades at this point. I'll give you a little bit of history in my part of the lecture here. But this is something, we have many papers, we've published a bit about this. But in principle, this became really, went from abstract to really possible back in 2014, which is Oxford Nanopore sequencing. Have any of you heard of or started using Nanopore sequencing? So we've got some people already in this space, which is great, because we actually will have a surprise also next week. There'll be some surprise samples every year for the course. We have a mystery RNA sample. Last year was induced pluripotent stem cells that will help us make woolly mammoths. And so next week we'll find out what the secret RNA sample that you will get to prep and sequence will be. Just to build some suspense, I won't tell you anything about it, but it's been very exciting. There's actually two different options we're looking at. But in case, it was very abstract to say, can you sequence a single molecule and examine it? There were early versions in the late 2000s that showed this was possible. But the first were high throughput nanopore system, again, some of you are already familiar with, it's called Oxford Nanopore Technologies. They took this concept of being able to have a single molecule thread through a pore and read out the bases, A, C, G, T here, you can see, as it's transmitting through the pore, usually at hundreds of bases per second. It's really science fiction, and even at the time, I put these two models up here because they didn't even know in 2009 or 10 what would be the best way to do this. So for example, you could pull a motor protein here, which you'll hear about later, and pull the DNA through, but you need something like a topoisomerase or some way to spread the molecule and pull through. Or one, actually, Oxford filed patents on both these ideas. What if you chop one base at a time, then watch it fall through the pore, and then measure essentially the signal of each base as it falls through the pore. So they filed patents on both. They tried to license patents, and but in the early days, it was not clear which one would win. But the one that did become commercialized, and the one that you'll be using in the course, is what was originally called strand-seq, and now they're just called nanopore sequencing. But quite literally, you're pulling a single molecule, threading it through a pore, and monitoring it. And they got a lot of excitement in the very early days, making something called the MinION, which is something you could plug in theory, put a drop of blood in, and sequence anyone, anywhere, and now some sorts of examples of that coming soon. But there originally was alpha hemolysin, was essentially the pore that would be pulled through, and it was just one little MinION, which was funny because at the same time, the Minions movie, did anyone see those movies, like little creatures and minions? And they would always say, no, it's MinION, because they're British, it's my accent, but it's MinION. It's not Minion, but at the same time, the movies were coming out, which I thought was just bad marketing personally. But the MinION, as they like to say, came out, and then later the Promethean, which we'll be using for this course. So Oxford Nanopore gives us some discounts to help get some reagents for the course, and you're going to learn about how to run and prepare samples and run them on the Promethean. For the British, said Promethean, maybe. Well, you're in England, Theo, so you can tell us if that sounds right. But so these are now the commercialized and fully functional versions of these Nanopore platforms, which again we'll be focusing a lot in the course. And they really enable some really exciting applications of technologies. For example, there's even a club called the long read club. Like, how long of a molecule could you get? Because a lot of the sequences I'll tell you about today were hundreds of bases, maybe getting to 1,000 bases, but they can get to 2 megabases or 2 million bases long. Was first achieved a few years ago. There's literally a website called thelongreadclub.org where people just hang out and compare what's the longest read they got. I think this is fabulous. It's a fun club. It's a super cool key club, but I think it's great. And the really very long reads indeed is if you see a read over 1 megabase, it's called a whale. And this is just in the lingo. You'll never hear about this anywhere outside of genomics communities, but if you see a whale, it's very exciting. It's like you're in the ocean of data and you see a whale. The idea anyway is called the long read club, and we're all looking for whales. But what's interesting about this is, well, several things. This is the longest way you can sequence today by far. This is in the millions of bases. The next closest technology can get you maybe into several tens of thousands. So it's really orders of magnitude longer than anything you can see, even with PacBio, which I'll show you in a little bit. And it enables really a lot of applications for many genomes. This is actually the size, it's a log scale, the size of, say, bacteria, viruses, archaea, or eukaryotes like humans. But you can see once you're getting into thousands of bases or millions of bases, if you can get several million bases, this covers like almost half of the tree of life you can just sequence with one molecule, which is just kind of extraordinary just to see in one plot. Now, 15 years ago, this was pure science fiction and fantasy, and now you can do it with a single molecule. So this is really beautiful illustration. Now, someday, maybe we can get to the point where you're doing tens, hundreds of millions of bases, then you can do whole chromosomes, and that eventually we get closer to doing the whole genome in one fell swoop. We're not quite there yet, and then actually, as you'll hear about in the library prep, the hardest part about getting DNA to get intact is making sure it doesn't break. Because you have naked DNA moving around a tube, it will fragment, it will break, it's unstable. And so that actually is almost a biophysical challenge more so than a technical challenge getting it before. But because of this technology, and there's very long reads indeed, the technology lets you sequence anything, anywhere because it's so small. So some of the examples of this has been Scott Tye, who's a collaborator for the laboratory of the Extreme Microbiome Project, and one of the co-founders. He went to Antarctica. Actually, the sequencer was still running even after the battery on the laptop died because it got too cold. So you can really take it anywhere. This is one example. This is some of the example of what he did in the Antarctic Dry Valley. See here. I've talked about this a bit. Just a picture of me talking about becoming a spacefaring species. And back in 2016, we thought, well, could we take it in microgravity? And actually, Andy Feinberg was the first person to say, well, let's try this on a parabolic flight. And you can see at the bottom of the tube, any of you do pipetting, that it's not what you want your tip to do. But he was trying. Also, there's people puking behind him. He's trying to maintain his composure. If possible, you're going to see it floating, tubes are going around, but you want to do some transfers on these pipettes, and eventually you get to the bottom of the parabola. Everybody's got fun, but he's smiling, he's maintaining composure, and we showed back then that it was actually possible. It passed the test. You can actually sequence in microgravity, or at least it worked. And then we published this back in 2016, so the nanopores appeared ready for liftoff. And then indeed, actually, Kate Rubens, who's one of our collaborators, and actually we're trying to recruit her to Cornell, hopefully soon. She was the next astronaut to go to the space station, actually, was a trained virologist. She used to have her own lab at the Whitehead Institute, and it just happened to be the next one in the queue to go to space. There's a fully trained scientist, had her own molecular biology lab, and she was the one to do the first nanopore sequencing in space. So then NASA, said, okay, sure, don't tell anyone. It's very exciting. I was like, it's on our embargo. And the next day they had a press release and they had this video come out as well. Let's see, play this video here. It's just her holding it close. They added that music. That was not me. Very dramatic music from NASA that you can see here. Getting ready. And you can see in real time of the data processing, which you will do in the course. But you can start to see the fragments as they're being pulled through the core and see the data being generated in real time. And so that was very exciting because by 2016 we knew it could go anywhere on Earth, anywhere in space with the very early versions of this. This is one of our missions we were very fortunate to contribute to called the biomolecule sequencer mission. Meaning, again, you can do things really anywhere, anytime. You can add dramatic music to your self-sequencing wherever you might like. But of course, that does raise some questions. So for example, how do you know you did it right? You saw, if you go back a little bit, there was some noise that looked like, because you're watching quite literally single molecules move through a pore, which is, you can see an electrical signal, but how do you know you did it right? What's the standard? For example, when you run a gel for an experiment lab, you have a ladder that tells you the size fragments. It's the standard, so you know what you're looking at. And for DNA, we didn't always have that, and it's even more important to have standards when you have new methods, and new methods need standards. And I like to say, you want to have this, ideally that would be not just a little small piece of a virus, but ideally like a whole genome in a bottle. And this was a consortium that started back in 2013-14 called the Genome in a Bottle Consortium. It's a little bit of a play on a genie in a bottle. Does anyone remember that show? I think, okay, a few people. Sometimes I wear my cultural references. I'm just too old now. So sometimes I'll put a movie in there and no one knows what I'm talking about. It's so fun. That's good. A few nods. But the idea was that this is a standard that's in the body. You take it, sequence it, and you know the location of every base, you know the modification. I'll describe some of the work we've done. This is it's through NIST, the National Institute of Standards and Technology, the Food and Drug Administration, so a collaboration between them and the National Cancer Institute to build these standards. And we need them on Earth and in space. And the thing we always worry about is that you're just getting together to make standards. you don't just want to have a meeting. I don't know if you've ever seen this little comic, but if you're lonely, are you tired of working on your own? Do you hate making decisions? You can hold a meeting, and you can see people and show charts and feel important and eat donuts. And meetings, some people consider to be the practical alternative to work. So this is what I often go to these meetings, and I have this up there to say, we're not just here to meet and hang out and have donuts. We should actually make standards. So I was very excited that this didn't happen, but we actually put them together, and they're now publicly available. And so you can go, you can order them from L'Oreal, you can get them from this website. It's a nonprofit, but it's just to have a sanity check for when you do sequencing. How do you know you did it right? They have this for the human genome as well as microbial genomes, and we'll be looking at some of these data a little bit in the course. So I bring this up because I'll talk about new technologies, which are of course very exciting, have new methods, new tools. You have to know that they're real and adjudicate fact from fiction. So this is the way we often do so in genomics. So to give you a bit more background on that, did anyone use microarrays? Anyone do microarray sequencing? Let's see, this is where I'm too old. No one did that anymore. I did my whole PhD on it. And now I've become that professor I used to make fun of, like, oh, he's talking about, oh, it took me 6 years to do this one thing. I can do it in one day now. But it happened. It's the inevitable march of progress. It's a good thing. But there was a time when there were different kinds of microarray manufacturers in the 2000s. I call it the early noughties, like not is a zero, and the early noughties, you could say, is the mid noughts, but no one says that. I'm trying to start a new cultural phenomenon where this is back in '93, back in '97. No one's going to do that. That's fine. But in any case, back in the late noughties, there were many arrays, and there were many companies making them. So they made samples again at the FDA and NIST to say, let's take the same two samples, send them to different labs, and we get the same answer. And that was the arrays. And then by 2014, though, there was actually for 2010, 2011, there were many sequencing companies, and this is essentially not all of your sequencers. That's why you're here. But one of the first real tests we did was take the same RNA sample and try it on many different platforms, including Illumina, Tag Proton. There's a personal genome machine, 4x4 and PacBio. I'll tell you a little bit about these technologies today, kind of the introductory lecture. And this came out in 2014, and then we did a follow-up on DNA in 2021 called the phase 2 study, which is again taking those genome and bottle standards and characterize them. And I'd like to say, because it was called the SEQC study, Sequencing Quality Control, and SEQC2 was the one focused on DNA, which then I'd say, my wife, what do you work on? I mean, that's like, it's sexy too, like I'm bringing sexy back, is what I'd say. And she would say, no, you're not. And I'd say, yes, I am. Anyway, it didn't work out. But what we did do is take these samples and say, we're bringing, adjudicating the best practices. And what's good is now the website has a Google group, an analysis team. We'll be using Collaboratory for this course, which Teo pioneered to make sure we can share the code. And also what's good is when you build your code for this course, you can then use it, analyze it, we can also see it. And so Genome and Bottle Group also has code sharing that's online. So again, the resources are online, you'll learn about some of them in the course. And then again, these standards are basically ways, you can see here some like the GitHubs and the repositories, which yeah, we nice. We just had some emails this morning about a paper from 6 years ago and someone looking for code, and then one from 3 years ago. It's very important whenever you build code for a class. If you think, oh, I'm just doing it for a class or for this one figure for one paper, make sure you keep your code well organized, because you never know, and 3 years later you'll be thinking, oh, I did write something for a class once and I could use that script or I could use that code. It's very good to keep it organized. I promise it'll be useful for the rest of your life. At least the concepts of it. The syntax might change in the language. For example, I have thousands of Perl scripts that I've written that no one wants to look at anymore. Has anyone ever written in Perl, that language? Anybody? No, see again. Oh, yeah, good one. Okay, good. Practical extraction and reporting language, and I have many scripts that I don't use the actual script, but a lot of the syntax or the logic of the scripts you can, of course, translate to other languages. So it's worth knowing. So again, these are the there's a paper on the standards. These are basically two trios, so a father, mother, son. The reason this is important genetically, because if you take one sample and you characterize it, you could look at billions of bases and see whether something was sequenced correctly. If you have a father, a son, and a daughter, or a father, mother, son, if you have a trio, you can look and see, is there something that's called Mendelian misinheritance? Is it something for if the mother has a G and the father has G's at one location, it's impossible for the child to have a C unless you have a mutation that happened like in uterus or as an embryo. You can look at cases of basically impossible inheritance and see either there should be zero, short for a little bit of noise, or again, de novo mutations, but they're very rare. And they give you, the trios are a nice way to also, give a really good accurate measure of the sequencing for one sample and also between a generation. So these are the samples. And the other few bits of points before I turn it over to Dr. Park in a second here, but we also have looked at these samples for an epigenetics profile, because you will also learn, of course, When you look at single molecule data, actually one of the best things about it, well, actually I just described it, but because you're looking at a single molecule that's not been amplified or altered, it's the molecules that say it came from your blood. It has methyl groups, it has other modifications to it, and you can actually see them when you pull it through the pores. So this is another data set and resource we'll be talking about is epigenetic supply control, which is again, you think about cytosine, and so this essentially, it's of course one of the four core bases of your DNA. But there's also methylcytosine, which you can see is just a small methyl group difference. There's also hydroxymethylcytosine. There's other variations of adeoxoguanosine. There's other many variations of these nucleic acids, and they have some epigenetic impact. They can alter gene expression. They can maintain gene expression going up or down. They can contribute to silencing of genes, which we'll learn about a little bit later. But what's interesting is when you want to measure this, you're really trying to see, well, it's just a methyl group that's different. So you think, well, I would like to measure this. One thing you can do is treat with bisulfite, and if you treat bisulfite, you can see here if it's methylated. You can see here that if you, this is the original molecule, you separate two strands of DNA, and you look at the top strand here and the bottom. You can see the methylated cytosines. They actually don't get deaminated. The loss of the amino group here, it eventually becomes uracil, so it's a slight change. But if you do that, if it's an unmethylated cytosine, you can see the C becomes U, and then after you do amplification, it becomes a T. So this essentially is a way we can see, if you sequence samples after you've done this chemical treatment, you can tell if something methylated or not with this chemical treatment. It's a very harsh chemical treatment to the DNA, but it's a way that's been around for actually decades to do this really relatively simple way to see if something methylated or not. It's interesting to note there is very rare cases of deamination you get, but it's hundreds or thousands of times less that you'll see this switch of thymine, but otherwise, most things that are methylated do not get deaminated, and they'll stay in that structure, and then you can see when you sequence them, you can compare the original strand versus what was after the amplification and the treatment, and see what gave the methylation at one spot or another. And this is difficult and hard, and they're actually on this paper, there's many methods to do this. You can see here you can do library preparation first, which you'll learn about and actually do, then do bisulfite amplify. You can do other steps of N-tagging or ligation to these molecules. There are a range of methods to do whole genome methylation profiling. But the one that we like the best and the one you're going to learn about and actually do is when you sequence the whole genome with a single molecule sequence, you get, of course, some liver mutations, but you get methylcytosine, hydroxymethylcytosine. There are a number of other modified bases that you can pull out from the data because it's the original molecule. So instead of having to do these harsh chemical treatments, which I put them up here because I want you to know that there are standards you can look at and there are other methods, but the method that would give you all the information in theory right out of the gate is actually doing it with a nanopore type sequencer or a single molecule sequencer. Just some more background about this. This is a chemical treatment. You see almost all these other ones are bisulfite conversion. That's how you add this part of the chemical treatment. There are other ways you can create deamination. There are actually enzymes called deaminases, which do exactly as the name describes. They take away the amino group. And some of these are ApoVec, for example, enzymes. And so this is just another example of a way you can, instead of doing a harsh chemical treatment, you can do an enzyme treatment to get the same effect. We published that in that paper as well as a way to actually affect the same change. And again, these resources are available. This is from the paper that just all the sequencing data that you can use also as a positive control for some of the analysis. And then I think we have, there's also, oh yeah, so here a lot more about this lecture 5. So just a preview, we're going to do a much deeper dive on the methylation profiling just in lecture 5. And I want to take a moment, so I think Jiwoon, who's next, and Dr. Park, if you'd like to do a quick intro for yourself, we're going to make it as weird as possible, so you have to introduce yourself. But it's a very nice picture. But take away for a little bit.
Yeah, thank you. I think we are now kind of switching and transitioning to introduction of myself and also Theo as well. Um, Quick intro for myself. I'm a postdoc. I work for Chris and also George Church at Harvard. I've been doing a lot of spatial and single cell integration, data generation, modeling. My PhD was also PBSB. I did a lot of liver pathobiology and how the regeneration really leaves the epigenetic marks behind, and looked at the single cell, like from experimental models like stem cell derived cells models, but also to computational like single cell and spatial. Um, and super happy to work with you guys too. And also a lot of the spatial will be coming out in the next few weeks or more of like a middle of the lecture series as well. If you can go to the next slide, please, Chris. So speaking of spatial, I think a lot of you probably heard about spatial omics already, and we've been pushing this course to also include not just like single molecule sequencing, but also spatial omics because we are now able to image those single molecules into actual physical space in our tissues. And spatial transcriptomics and proteomics has been methods of the year, um, actually in 2021 and again in 2024 with the proteomics, and has been one of the technologies that is going to be very useful for all of you, especially if you want to pursue research and especially if you want to put more clinically relevant research as well. So this is one of the examples of like how we can show and map these individual molecules, RNAs or proteins into 3D space, which you can down after segmentation and clustering and all of these analysis, you can go all the way down to, as you can see in the middle, right, like the bottom cell labels and niches, you can visualize your single cell data essentially, but and all the sequences onto an actual XY coordinates of like how it's actually organizing a network of tissues. Can you go to the next slide, please? And these are some of like the preview of what you will get. Like we'll definitely spend a couple of lectures trying to run through different methodologies, how does technology work, what are the chemistries behind, and how do we analyze some of the data sets. But just for your reference and for your future research, these are some of the spatial sequencing, but also spatial imaging capabilities that we have at Mason Lab, also at Weill Cornell as well. We heavily use Cosmics, IMC, and Cellscape, more so on Cosmics because it's one of the highest resolution, but also it allows us to do now. We're still optimizing, but it allows us to now do the spatial multiomics, basically have H&E, RNA, and protein on exact same slide. But we also have a plethora of different technologies like Codex Xenium, Visium, Cereusig, and Pixelgen. And there are a couple other more spatial technologies that we're currently piloting right now. So if you guys have any questions, I think Dr. Mason and myself can be a very good resources that you can reach out to if you ever want to use it for your own research as well.
Thank you, and thanks, Dr. Parker. Also, some of the papers we'll be talking about in the course, she's characterized a lot of these technologies. You'll hear a lot more detail of this in the later lectures and their applications, and also a chance to play with some of the data. And some of them are really bleeding edge. Some of them just come out in the past 6 months, but are really interesting ways to visualize internal changes inside cells and the proteins and the transcripts, and also other imaging data. So it's a very exciting time in the spatial imaging side. And so thank you. Also, one fun fact about the IMC, the imaging mass cytometer, we have two of them here at Cornell, the Precision Medicine Center. And inside the machine, it's hotter than the surface of the sun, just at the point where the laser is. So there's a little fun fact. It actually violated also the LEED certification for the research building because it had so much exhaust. It's no longer LEED certified. But anyway, that's great, beautiful data. It just is very hot on the inside.
I didn't know that. Yeah. One more thing. We will be using some of the astronaut samples for the spatial data as well. So
Yes. Exciting new astronaut data for some of your homework. So we try to make the homeworks as interesting as possible and as weird as possible, which are the two main goals. Okay, so next up, Theodore was a Churchill Scholar and incoming XMB PhD student as well as helped design and be part of the course. And Theodore, do you want to do some quick background on yourself?
Sure. Hi, everybody. Funny enough, I'm in Cambridge, Boston today, not Cambridge, England, but usually I'll be in Cambridge, England. I'll be virtual for the duration of the course, so always feel free to reach out to me per email. I'm a bioinformatics specialist in the Mason Lab. If you go to the next slide, you can see me in action. Basically data gets produced and I analyze it, mainly our Nanopore long-read direct RNA sequencing data. And yeah, this is my second year teaching this course, and it's really exciting. I think we've, made it a really dynamic and engaging experience, and I'm looking forward to an even better class this year. So thank you all for joining us, and I look forward to working with you through the course of the semester.
Thanks, Theo. Thanks. Just credit, this was a class I've been brewing for a while on campus, but really rejuvenated a lot of the hands-on parts. We wanted people to learn the code better, to learn the preps, and actually be able to take these tools and bring them back to your own lab and use them in theory for the rest of your life. So with that, we also have a quick survey, again, a quick break of what What is not single molecule sequencing, and see if you can take a quick survey, so you can get a little bit of what is or is not SMS. But I want to, single molecule sequencing, did not really drive the last 10 or even 15 years of sequencing revolution. But it is important to look at this lecture. How many of you have seen this slide before? Anybody? Almost everybody. Of course you have, because this is the most overused slide in every genomics course. But the reason it's overused is with good reasons. That this represents the fastest pace of technological change in human history. You can see Moore's law, which is a doubling of transistors every 18 to 24 months on microprocessors, was really an extraordinary log linear increase in density consistently since the 60s, which is extraordinary to see. That's why your phone today is more powerful than supercomputers in the 80s. We just take it for granted. We don't even think it's that magical. It's just normal. We're just texting or looking at TikTok videos. But it is an extraordinarily powerful machine. And that was what enabled a lot of our current technological era. But and you'll see, again, here we are in the mid-noughties, which we'll talk about. There's a lot of froth and competition and battles with new patents and methods to do sequencing and do it in massively parallel scale, called MPS, massively parallel sequencing. And this led to the cost, instead of being hundreds of millions of dollars to sequence one human genome, it's come down to tens of thousands, or these days even into the hundreds of dollars. It's the reagent cost to do a sequencing of a whole genome, which is really an extraordinary dynamic change. It is literally the fastest pace of technological change that we've ever seen in human history. I don't know if this will ever be beat again, just because it was such a dramatic speed up of technology. It's worth actually noting, and George Church, we were just talking about, he had this really great slide back, from the mid-noughties, is that at the current, if you just had kept pace at Moore's law, it would have taken us till about, maybe 2020s to 2040 to get to $1,000 genome, and we had eclipsed to beat that well before that because of the technological change. And so you might, so it's exciting is, in 2014 we thought we'd get to $1,000 genome, and there were other headlines saying, oh my gosh, we're gonna get to a $100 genome. Fun fact about this headline is Illumina announced this in 2017, saying we're going to have a $100 genome soon, and then their stock went up about 20%. Everyone was terrified. They didn't know what it even meant. They don't have it today. It's just amazing. If you want to get everyone excited and raising money in the stock price, that did work for them, but eventually they got eclipsed a bit by other sequencing providers. I'll tell you about today. Ultimate Genomics, one that did get it to $100 or even less. And you can see here, actually, it's now down to $80. They just announced this in February that they can do the cost of reagents with their Solaris, it's called. They can get down to $80 for a genome, which is really extraordinary. Like, it's now, I keep thinking in my head, at some point the cost of a New York City cocktail, which keeps going up, will be more than a genome. And at some point you'll just leave the bar and be like, to hell with it, I'm gonna go sequence genomes instead of drinking. It's better for me. I'll learn more. Stop drinking. That, at least that's what I'm in. I don't know what you'll do, but it's getting really cheap. We just got even a quote today for $30 a sample for some shotgun sequencing. So it means you can do hundreds or thousands or even millions of samples worth of data because it's getting so cheap just to do one genome. So very exciting, but and we'll get into what you do with the data during the course. But what's interesting is that's, and I'll walk you through some of the other technologies that got us here, but it's worth noting that will probably be the next 10 years, that was the last 10, 15 years, is that the single molecule sequencing, and this is again another example of how it happens and how it functions, and you see these differences of the current picoamps as a function of as molecules move through the pore. You get the unmodified base or modified base differentiation. You can see both, and you can see it very fast. We'll come back to Roche in a second, which just released a new technology that's even faster. But these are it breaks down generally into kind of these old school methods or new school methods. So that's single molecule, but I want to walk you through what were some of the not single molecule. What that means is you're going to have either hundreds or thousands of templates that you're looking at the same time just to get a signal from this. So I'll walk you through what that looks like. But there was Sanger sequencing. There's also, has anyone done Sanger sequencing? Of course, probably. Okay, a few people, some validation. All right, good. But generally rare today. And there are other methods of these more new school. This is also a really good trip hop album I recommend called Old School versus New School. But Sanger sequencing was, what Fred Sanger won the Nobel Prize. 2 Nobel Prizes actually, he won. But one of them was particularly for the ability to do, and the concept of doing Sanger sequencing, where you actually have we do a PCR, an amplification reaction with chain terminating dideoxynucleotides or nucleotide triphosphates. So you can't go past that one base as you're kind of doing the reaction. You basically take a mixture. You have the original. You can see a sequence on the left. You have a mixture of DNA and DPs, and some are fluorescently labeled with a different label for each of the 4 bases. And then as they get mixed up and then put down in a gel, you can see based on their size fragment where they have a different fluorophore, so you can see this burst of color that tells you which base you have at which size fragment. And this gives you a way to get in the ranges of hundreds, even several hundred bases, but then it would start to get noisy, and this was the standard. It wouldn't work past a few hundred bases, but this was the standard. This is what led us to sequence the human genome, the very first human genome. This was the standard, really for decades. That was the best we could do. We could increase the efficiency on these chromatograms, but we couldn't have high throughput scale effectively as a Sanger sequencing. But what happened again in the mid-noughties was actually Illumina acquired a company called Solexa, and they had a different idea. So instead of doing mixtures of molecules and getting a one molecule basically, because if you go back to this, you're really getting just a synthesis view of one molecule. If you see a heterozygous, because you have half your DNA from your mother, half from your father, and you get a heterozygote, if it's 2 different bases, you see a second peak. But you really just have one molecule. Like, to get all this work to get one molecule, it's a few hundred bases. If you repeat that again and again, billions of times to sequence one genome, it's expensive, it's laborious, computationally difficult. But what Illumina did is acquired a company that had perfected this idea of, okay, wait a minute, what if we take a fragment of DNA and we just prepare the sample to have something on the top and the bottom, basically ligate these adapters, so you do a ligation reaction, or you do various ways to do it, but DNA ligase is a common way. So you have a different top and bottom. And then if you have a cluster, basically a small flow cell, which we don't have here, but we'll bring it to the next class. And it's just like, imagine a swimming pool. And if you dive into the pool, but at the bottom of the swimming pool, there's 8 lanes, you can see all these little nubs at the bottom of the pool. And if you, as sort of a molecule, are diving through, and if you basically stuck onto the bottom, you can imagine yourself making an arc at the bottom of the pool. And if you make like kind of a handstand, which is kind of what the molecules actually do on the flow cell, you then have a cluster. You got one template, one piece of DNA that's bound, and then been copied with one cycle of an amplification. And then if you repeat this for several cycles, you'll grow a cluster. So you go from one molecule, then dozens or several hundred molecules in these clusters. And again, it's like having a bunch of people at the bottom of a swimming pool standing up that have been cloned of each other. And then when you do that, you can actually then do this sequencing. It's called sequencing by synthesis in massively parallel, because you basically take, you do an incorporation reaction, you image that, and this is what the flow cell looks like on the lower left. You can see all these colors. It's one color per base because there's 4 bases. Keep it simple. And then it's a fluorophore. And then you cleave it and you repeat it and you wash away and then go to the next reaction. Cleave it, do the next reaction. You keep repeating this. And what you're doing, you're synthesizing the complementary strand essentially by this synthesis and sequencing process. Hence, it's called sequencing by synthesis, or SBS. And if you keep your eye in the same spot, you can see it's like basically stacking bricks. Each one of these reactions is kind of like an inverted brick stack, where you can see each one of the bases. If you look at the exact same location every cycle, you then get each one of the colors, and that tells you which base you have. And so that's a way, because the reason it's a cluster is you want to get enough signal. You can see those clusters are not all perfectly the same size. They're circles, but some are smaller, some are bigger. But you basically grew a cluster, and you image it every cycle. And when you stack these basically bricks of bases, you then get the readout, and you can say, okay, I have now read out this one reaction at this one spot. And first they had tens of thousands, and today they have billions of those little spots in one reaction. So then you can get basically terabases of data within 24 to 48 hours from this kind of chemistry. So fairly simple concept, but then it gives you a lot of data. So this is, what the way this is done in sequencing by synthesis was also behind several other technologies in the mid-noughties, where they have these block reversible terminators. Here's the nucleobase, and you can see this is, a normal nucleobase would stop about here. Here's the sugar. There's some of the purines or pyrimidines. There's the triphosphate group. But the reason these are so interesting molecules is they have this large side chain and a huge fluorophore, so when it sticks there, you can image it. But it's a weird molecule. Imagine if you're a DNA polymerase, you're expecting this as an enzyme, and suddenly this shows up, and you're supposed to process it, analyze it. I'll show you a movie of this in a second. It is a weird molecule you have to deal with, but Helicos did it, Illumina did it. They built these chemistries that you actually mutate the polymerase so that it is tolerant of that. But so this is done twice for these 2 companies. And originally they came out with 4-channel chemistry, but then they realized there was some noise in the G was one of the noisier ones. They thought, well, what if you think about 2 bits, that gives you actually 4 pieces of information. So if you see both colors, that would be an A, P is green, C is red, and G is dark, and that gives you two bits, gives you 4 types of information, 0, 1, 0, 1. So it's okay, we can do 2-channel chemistry, and eventually the 2 imaging cycles, you get 1 channel, 1 color, And then 2 cycles is also going to be 4 kinds of bases. But they've iterated on the kinds of chemistries. They're called 4-channel, 2-channel, or 1-channel. But basically, the concept is relatively the same. But you have to have something be labeled, or in the case of G, dark, differentiate whether you have a base there or not. And so that's most of the Illumina chemistry. There's a few other variations of Illumina, as they also do paired end sequencing. So that this, if you look at this, it looks like, oh, there's a single molecule that seems straightforward. But you can also do something called paired end sequencing, where you do that same cluster amplification, and you cut there, and then you linearize it. So you basically do read 1 down, then you resynthesize that strand, and then you actually then linearize it again and cut there, and then you sequence down the other direction, and that's read 2. So you will have some data sets that are paired end sequencing. And what that means is you've read from the top and the bottom of this molecule, and they're usually about a few hundred bases apart. So when you see paired end sequencing, it means you just got, well, it's different pairs of the different ends of one molecule that were sequenced. So you'll see some of this when you read the papers and see the literature, and that's just basically how it gets paired. There's also a video of this if you want to take a look at that for your background. And the other cool thing about paired end sequencing is you can then also start to add in indexes. You can do this for other sequencing too, but you can think about, well, if you can have, this is what you're trying to sequence, right? Say it's my DNA, your DNA, it could be a bacteria, it could be anything. But if you start to add in an index, so you have these different paired end reads, but you add in this small barcode, like 8 or 10 bases that are unique to that sample, then you can do 100 samples at a time, 1,000 samples at a time, as long as you know that this is a unique index. That you basically imagine a plate and you have 96 different samples, add a little barcode each well before you actually pull them, and you can use computational tools to say, oh, this came from that well or this well based on this little barcode. And so here's indexing, or this basically way to do multiplexing. You'll also see this some of the tools and homework, and it's now very standard in the field, but it was a fairly simple concept. You'll just add a little bit of DNA specific to one sample in a little well, a little plate, and that lets you differentiate that computationally down the road. And also feel free to join with questions, any other questions. That's Illumina, which is still probably 80% of the market, 85% of the market is Illumina. It is the kind of the gold standard. It's the most commonly used sequencer. And that had been the case really for 5 to 6 years. But in the last 2 years, it's gotten a bit more competitive. There's new technologies that have come, new methods. One of them is PacBio has been around for a while, but it's gotten more higher throughput. Let me see if I can get this here. Actually, I'll get the link here. I will show you. There's one that has a very dramatic music. The soundtrack.
Introducing the PacBio Sequel 2 system, powered by single molecule, real time.
Not that I want to give you an advertisement, but they spend millions of dollars on animators, so we might as well use their animation because they're often pretty good. But this is one of the later versions of their sequencer.
Sequencing technology. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a smart bell library is created by ligating adapters to double-stranded DNA, creating a circular template.
Quick note, very smart advertising. Call all of your technology smart. It's very clever marketing. Smart bell, smart library, smart sequencing. Just clever note. But you can see this, the double-stranded DNA and their specific adapters that they make. Illumina just have them as a single base. Their adapters are loops, which is unusual. But you'll see why that's relevant to this second. That's interesting.
Primer and polymerase are added to the library that is placed on the instrument for sequencing. At the core of smart sequencing, it's the smart cell, which contains millions of tiny wells.
Smart cell, it's smart, very clever.
It's called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel 2 system, you can optimize your results with 2 sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads.
One thing about this is very, really smart idea that, pun intended, a smart idea where essentially it's a circularized template, and you go through each. Polymerase is what's called strand displacing. So even though it's a double-stranded molecule, it'll keep pushing the other strand out, and so you can see the same template many, many times. What's interesting is the polymerase, the first pass accuracy is only about 90% accurate, which is not that good. You think, well, I want to be better than that, but because it's almost perfectly random, if you see the same base, say you have a 10% chance of being wrong here, 10% chance of being wrong here. By the time you see the same base 4 times, that's really, 1 1/10 to the fourth power. So your chance of being wrong by the time you see something 4 times perfectly random is really, one in 10,000. You chance of being wrong, or 1 in a million. If you sequence enough times, it could be 1 in a million. The odds get better every time you see it. So this is something called CCF reads, or what they now call hi-fi. Also, fun marketing trick. It used to be called CCS, and now they call it hi-fi. It's the same thing. If you see old papers, they're the same thing. It just means you've seen the same template multiple times.
Or use the continuous long read sequencing mode to generate the longest possible reads.
And you can see here, they did say quickly, half the reads are more than 50 kb. So they get to 50 kilobases or even 100 kilobases, it's 100,000 bases, which is pretty long. But to date, have never gotten past 1,000,000 bases. Some of it's just those molecules are too hard to keep into a form. If you go back, if you think about some of the stoichiometry and the biophysics, these are molecules literally sitting at the bottom of a tiny little well that are kind of pooled into these smart spoilers. So it's kind of amazing to think about. They get down there, they're stable, and then there's a polymerase at the bottom that is pulling, moving us through and is happy enough to survive down there. Their polymerase, they also had to heavily mutate before it would survive. So if you're an enzyme, you're used to just doing processing DNA. You're not thinking I'm going to be at the bottom of a well where I'm stuck down there and pulling my DNA through. So it's really kind of a cool technology that they managed to get to work, but it did take them many years and many mutations to get to work. So that's PacBio, and that's there's a couple other videos here if you want to hear some of their original videos. One note about this, actually, their original one. Oh, that's the second one. Go back to their original. And what happens on the bottom? They do have a fun older video that talks about what happens at the bottom of ZMW. Is that it has the world's lowest detection volume. It's actually similar to, if you're ever looking at a microwave and you think, if I put my face to the microwave, how come my face doesn't get burned? It's because the wavelength of microwaves is much bigger than the hole of the aperture of the holes. But you do get a tiny bit of what's called an evanescent wave. So in this case, there's a laser light below each well, and you get a tiny little bit of a detection wave that comes up here of what's called an evanescent wave. And they can detect down to 20 zeptoliters, 10 to the minus 21, as an actual scientific notation. So next time you go to a bar, you should say I have like 2 zeptoliters and see what the bartender says. But it is a very small volume. It's like that very small part in the bottom of those wells. And so there's another video here, just link in the slides, where you can see how they get this immobilized template. And then, the other thing we'll come back to is because it's single molecule, you can tell if this template is cytosine or methyl-cytosine, regular base or modified base. You can see that. And so that's PacBio. PacBio's been out actually since 2009, and they've gotten better and better every year. So it's one other sort of just kind of cool technology to know. But there are other. Let's see this one here. The other one that's just come out was Roche, which has been working on this for about 15 years and just announced it actually last month. It's called sequencing by expansion. For whatever reason, most of the videos in sequencing have a British woman speaking. I'm not sure what that means. Maybe we all take comfort in the authority of a British accent. I'm not sure, but it is. I think we'll see it here as well.
DNA bases are separated by only 3.4 angstroms, the width of a few atoms. DNA sequencing methods cannot easily resolve this dimension. Our sequencing by expansion technology, or SBX for short, overcomes this limitation by creating a surrogate molecule called an expandomer. This molecule is more than 50 times longer than its target DNA and encodes the DNA sequence information in large, high signal-to-noise reporters. The building blocks for the expandomer are novel nucleotides called XNTPs. Expandomers are formed in a modified DNA replication process where XNTPs sequentially link along a target DNA template using a proprietary enzymatic process. This forms a double-stranded helix. Each of the 4 XNTP types, depicted as A, C, G, and T, has a tether that is linked between the base and the alpha phosphate.
And the alpha is the proximal phosphate, but you can see at the bases, they almost look like little people with parachutes. Which is, again, think about, I want to design a sequencer, I want to design a molecule. It's just a fascinating chemistry to think, I'm going to make these really unwieldy giant constructs for nucleotides. Now, normally if you'd asked me 20 years ago, would this work? I would have said, this is a crazy idea, this will never work, this seems hard. To be fair, it did take Roche about 15 years to get this to work well enough that they just announced it this year. But you can see it just chemically, it might seem complicated, but the reason they've done this is normally You know, enzymes in your body are built to work very fast. And so if anything, when molecules move through pores, it's too fast. But this helps slow them down.
Live signal noise reporter that mirrors the base sequence identity. As the extension proceeds, only an XNTP that correctly complements the next base on the DNA template will be incorporated. The sequence information of the DNA is now expressed in the reporter sequence of the XNTP. This cycle of linking XNTPs continues sequentially and extends along the entire length of the DNA. After synthesis, a reagent is added which degrades the DNA template and breaks the cleavable bond between each of the XNTP bases, allowing the backbone to expand. This product is called the expandimer. The expandimer sample is now enriched for high-value, long-read lengths. Stratos Genomics will capitalize on 2 decades of nanopore development that has attempted to directly sequence native DNA. By scaling up size and signal of expandimer reporters, SPX technology now enables high-accuracy nanopore-based DNA sequencing. In a nanopore detector, an expandimer chain is threaded electrically through a biological pore. As each reporter passes sequentially through the pore, the electric current is impeded, providing one of 4 signals that uniquely identifies the next base in the DNA sequence. A single CMOS array chip can measure thousands of nanopores, leading to sequencing rates in excess of 1 million bases per second.
Which is the fastest by far throughput of any machines. This was talked about for a while and just finally announced actually a month ago. So I mean that's millions of bases per second you can measure, so you can do billions of bases in a few hours. It's really terrific.
Stratos Genomics brings you the basics of life.
That's their tagline. But it's really, so this is something. So Stratos is work a lot on actually that ligation, that sort of expandimer. And then there was a company called Genia which made the pores, and so basically rotated apart and both put them together for the first time just this year. So this is very new but very interesting. We're working on ways to try and get one of them set up here hopefully in the next few months. So we don't have this for maybe this year's class, but then next year we have this also as a way to look at some of the sequencing differences. And then 2 other ones I want to tell you about this are sequencers you should be aware of. They're on the market again. As you can see, there's many ways to do high throughput sequencing, and these are the ones to think about. These are in that case, that was a single molecule sequencer, but it does have to be modified a bit before it can get ready for it. But BGI is the Beijing Genomics Institute that also has their own version. It's very similar to Illumina sequencing, and they have something called the T7, which when this first came out, it reminded me of the T1000, the Terminator. Do you remember the Terminator T1000? And I emailed BGI and they didn't find this funny, but I thought that was the first thing I thought was T1000. But it's not that. But they do have interesting talks they'll give. Where they have this was actually at the conference a couple of years ago, the same conference of in the future you can have rows and rows of these sequencers, and you can have it looked a little bit like the Terminator. I just thought it was interesting that they had this vision, almost near dystopian view of the future with reams, and they could do sequence everyone at birth, which I actually like that idea. But there was a question of what are they doing with all this data and where's it going, and that was not clear for a while. There's some unauthorized testing that had been being done. So there's a lot of questions about that, but at least as a goal to get that point, to have that much data, is something that was really hypothetical and ambitious. But with things like the Roche sequencer or some of the high throughput sequencers, you could actually get to the point where you could have everyone sequenced at birth, and everyone even over 50 years, 60 years, have a continual surveillance of them to see if they've accumulated new mutations. So the cost has finally come down, and the throughput has gone up. Where this original very aspirational goal is actually very much possible. It's kind of fascinating to see the progression. And then there's a new kid on the block, which I'll highlight here, is something called Avidity from Element Biosciences. So the last one to tell you about is the Element system, which again, they had in this video. One fun thing is they make it look like the DNA is like shooting out of the flow cell. I don't know what this means exactly. It's very exciting. It feels like it's like a fountain. If you ever get a chance to go to Jet Propulsion Laboratories, you should if you're a taxpayer, because your taxpayer is funded. They actually have a nice little imager of every piece of data that goes from any satellite in our solar system or outside our solar system for Voyager. And when data goes in or out, there's a little light that's at JPL. It's a very cool. If you ever get to tour, I recommend looking for it. It looks a lot like this. But this does not actually happen when you're in the lab. You won't see lights of DNA. I think it's very evocative though. So this is the first, and then we'll see the thing. It's called the Avidity.
All the elements of a sequencing platform reimagined. Avidity combines flexible, simple workflow and best-in-class data quality on a desktop system.
And we'll talk about this later, but these Q values are very important. You'll learn them a lot in this course. This is the quality. So it's Q30. So Q20 means it's 99% accurate. Q30 is 99.9% accurate. Q40 is 99.99% accurate. So it's a log scale, which we'll talk about more in the later lectures. But this just means you're getting it highly accurate. And you can see here's in paired end sequencing 2 by 150, and so it's kind of limited. And looking at how many reads you get per pulse. So when they announced this, this is sort of their competitive product. These are all generally good metrics.
Perform affordable genomic science at your bench. With our independent dual flow cell random access format, you can perform multiple experiments concurrently or independently. Avidity is built upon Element's new proprietary methodologies in chemistry, imaging, and data analysis. Low binding surfaces reduce background to maximize contrast to noise for the highest quality DNA sequencing data. Seamlessly compatible with existing library prep workflows, Avidity rivals leading sample input requirements. In addition, new surface amplification strategy decreases bias, eliminates amplification-based index crosstalk, and facilitates unprecedented data throughput on a desktop system. Chemically engineered avidite nucleotide substrates in an engineered polymerase decreases substrate concentration requirements by 2 orders of magnitude, overcoming sequencing cost barriers.
So this is one of the big innovations. These avidites is a combination of a polymerase plus this other molecule that binds to the templates, and so you get a lot more, in theory, a lot more signal and less noise because the flow cell's also been re-engineered, so the base has less noise. But this is something, and they also amplify what's called rolling circle amplification, which I think we should be in the next slide. But they amplify a lot of the targets, so you get a lot more DNA and a lot more signal from that original molecule. So this is another example where it's not single molecule sequencing because you're amplifying and making many copies of it to get more signal, but still is a very interesting chemical way and an idea to, I think, amplify signal.
Using the amplified DNA templates, our proprietary enzyme selects the correct avidite, providing exceptional accuracy. Hundreds of avidites bind per DNA feature, generating a scalable signal to maximize contrast to noise. Supporting both paired end and dual index sequencing, Avidity generates an output compatible with existing workflows and analysis pipelines. Element's innovations in modern optics and data analysis deliver the highest throughput available on a desktop system.
Yeah, the rest of this would have been added, but then he also said data, which I kind of like, that's a good language, that data. And so this is, this chemistry, next slide. Again, these are these avidites, these basically large, you start with one template and you amplify here, you can see this large amplification product. So it just gives you a lot more signals. So when you're looking for a chemical reaction, you have many copies of it. So it's easier to see. It's a fundamental idea. It's done through cycles, just to walk you through what you kind of just saw. There's a hybridization of the DNA to a circular template. You make these polynes, which is what's through rolling circle amplification. An enzyme called phi-twin, it just makes it many big copies like we just saw. And then the avidite is where you have this dye-labeled polymer that has multiple nucleotide arms that have the same nucleotide base. So it's really just a way to get more signal. And what's interesting about this is if they separate out essentially this amplification process from the point where you detect it, so they're decoupled. Whereas in PacBio, when you see the base, you're sequencing the base. Like for example, that's happening at the same time, so you have to get that spot on. Whereas here you can actually optimize this step, wash things out, and then detect what's left over, and then cleave that and move that on. So kind of making different steps ostensibly here, and I described to make it each one a little bit better. And so this can get through up to 600 cycles. You can see the bottom, so it can do this about 600 times before it starts to lose some signal. So we don't have one of these instruments here, but there's one at Sloan Kettering right across the street. And we've been using some of this data though for our major paper in the past couple of years. But that's it in a nutshell. Is the Element system, or the Element Biosciences, the company in the V24 is their recent system. And you'll hear a bit about this later, because actually this flow cell, this image, you can imagine this happening. We'll hear about this in the later lectures. But if you think about what this is, you have a system that does fluidics and optics and manages individual molecules. So it's great for sequencing, but you can also image what's happening on these flow cells. You'll learn about that in the spatial lectures, Whereas some of these systems like the Element, the Illumina systems they're not just sequencers, they also do imaging of cells or spatial transcriptomics, which you'll hear about again in later lectures. But it's really a great application of a complex imaging system that does a lot as well for fluidics and tracking fluorophores. So the last thing I'll mention before we jump to another quick break point is every platform. This is true really for any technology. I think these are all sequencers, and there's many kinds of sequencers in the market. So I think it's evidence of a lot of interest because you can have this be a substrate for precision medicine, for better oncology, better infectious disease. Sequencing at birth is really exciting. But every platform, whether it's proteomics, nucleic acids, with any detection molecule imaging, they all have various noise and various kinds of noise and thus error. So you'll sometimes hear about dephasing, which means a lagging or leading strand. So let's say you think you've incorporated one base at a time, but you actually grab two. And so sometimes you can't see that with the camera, but it's what's called a dephasing. Or you think you've incorporated one base, but you actually, the reaction didn't work. No chemistry is perfect. Every enzyme will have, even if an enzyme is 99.99% perfect, if there's billions of spots on the flow cell, you will have places where you think you incorporated one base, but you didn't, or you did two instead of one. It's called phasing errors. You can also have dark nucleotides. You think you have a fluorophore attached to a base. What if it falls off? What if it breaks? When you synthesize the nucleotides, again, even if your chemistry is 99.999% perfect, if you have billions of locations and reactions, you will have some dark nucleotides where it just falls off, it breaks, doesn't work. Every polymerase has an error. It doesn't always perfectly end with full fidelity copy correctly, and it's a range, so they're all perfections. And there's also just challenges with single molecule. Because you're looking at a single molecule, it could be noisy. If in a polymerase, the thing on a piece of DNA could be wiggling basically from the tail. And then every platform has its own specific errors. So there's all things to keep in mind when you're thinking about the kind of sequencers. And then I want to ask, besides, have you ever done anything with any other single molecule sequencing? If so, anyone, you signed up for the course, you must finish it. Has anyone done anything with other single molecule sequencing of any kind, proteins? Well, good, then you'll learn a lot, because no one has time yet. But there's other variations of looking at a single molecule. You can look at a single protein. We'll talk about this later. For example, quantum is one system. You can image very specific molecules on the flow cells, and you'll see some of this as well. So there'll be other variations of this. And then I think now I want to hand it over for a quick, quick adjustment. Let's see. Yes, we want to get you to have this good application and translation potential. But now also Theo, I think he's going to jump in this part.
Yeah, sure. So in terms of the last slide, I guess a really big emphasis is that what we're doing in the lab and the kind of research and the kind of sequencing analysis that we're doing really informs the work of this course. So understanding, right, and taking what is the cutting edge and turning it into something that's teachable. And that's something that you all help us with. And that means that since it is the cutting edge, it's something you can contribute to in terms of research products. How we want to sort of approach this content is that there are many sequencers that we just went through. It would be hard. There's no quiz right now, no quiz to like test your knowledge on every single one of them. But in essence, right, all of those we reviewed as concepts, which is sort of the pinnacle here in terms of the level of abstraction versus the amount of detail. They're all presented as concepts because they're important to compare to each other. There's certain technologies, so specifically Nanopore and some of the spatial technologies, where we're going to go much deeper in terms of the amount of detail that we're going to learn and versus the level of abstraction. And that means that you'll be working with the actual Nanopore data, programming, working with it hands-on, and eventually getting into the lab and preparing these sequencing libraries. And at the end, working through a final project. So as a part of this, right, there'll be weekly assignments modulated around coding and the support and homework keys on a weekly basis. So next slide. Last year, we tried to organize the course as sort of discrete topics and sort of thought of it in like the context of the general framework of the central dogma. But this year, next slide, it's a little bit all over the place. So DNA, RNA, protein are going to be integrated with each other more tightly. So you might have one lecture on DNA and then in the same session, another lecture on RNA. I hope that'll be enriching for everyone. And that's sort of the way our guest speakers and our topics prepared out. But since RNA really serves at the interface between DNA and protein, and because your main wet lab project will involve direct RNA sequence, Nanopore direct RNA sequencing technology, the RNA piece of this predominates a little more over the DNA and the protein. But at the start here, we have an introduction. So your first homework will try and provide some introductory CS experience, some introductory problems. And at the end, right, you'll be like Superman down there. This is actually what came up when I asked ChatGPT to visualize Chris Mason. This is yeah, so this is where you'll be at the end of the class, and you'll be presenting your own projects, analyzing your own data, thinking about a novel issue really deeply. So next slide. And yeah, and you know, a scientist, right, goes from every single layer and every single experiment. So that's why this class tries to take you start to finish through a sequencing experiment from the sample preparation all the way to final analysis and figure generation. So it's very exciting. It has wet and dry lab components. Next slide. And lastly, I like to sort of highlight this from the syllabus. We'll send out a text syllabus with the recap. But basically, you know, we allow, except for that pre-survey, since we want to know like what you know, but for the rest of the course, we allow the use of ChatGPT and think it's a very good tool for in-context learning. So if you look at the homework problems, if you look at the coding problems, and you think, well, I might not have a lot of coding experience, or I've never tried this, or I've never done it before, you can basically use ChatGPT as your own personal tutor to think about these questions, to think about these challenges. And I think it's a really, and even more and more so, I'm thinking of it as a really cool research paradigm for elucidating my hypotheses, for adding extra context behind it, even right for all of the biology that you may or may not know. So hopefully this class, in addition to all of our other stated biological goals, will help you learn how to use ChatGPT better. Next slide. And now we'd love to hear from all of you and meet you. So if you could take a minute, we'll go through the class, but say your name, your program, why you decided to take this class, and whether you collect magnets, and if you have a memorable magnet. Yeah, so next slide. So as you'll see, we're going to go back and forth between single molecule and this sort of single cell spatial world. Again, back and forth, back and forth. There are a lot of different questions that you can generally ask, whether they're exploratory in nature, whether they're hypothesis driven. A lot of the questions and ideas, the projects that we are going to engage with are mainly exploratory. And in terms of hypothesis driven or validation driven experiments, those won't be touched on or covered as much. But at the end of the day, optimally, right, you would get to the point where your sequencing method, you would have the standards, you would have the resolve, where your sequencing method would go from something that was just considered an exploratory technique to being used in a hypothesis-driven way, either because the cost has gone down so you can perform the necessary replicates on the experiments or otherwise. So next slide. So yeah, single cell, single sit, or single molecule versus bulk, right? So a lot of the technologies that Chris talked to you about are bulk sequencing technologies, which means that you amplify the molecule in some way in order to measure it. And that is really different from being able to measure something as a single molecule and to be able to understand it natively. And one of the sort of hardest concepts to wrap around, and something that we will bounce back millions of times within the program, within the course, is this distinction. But maybe the best way to think about it is to imagine that you're back in 1665, your name is Robert Hooke, and you've built this microscope, and you look through it and see the very first single cells. And you see this, and all of a sudden, right? Nobody's ever seen a cell before, so they probably had all kinds of assumptions, right, of what this looked like. But you see the first cells. What are the questions that you're going to ask of a single cell? Is it rectangular? You know, on the left, it seems like there's some rectangular stuff. Is it circular? What does the shapes actually mean? What are the different structures inside of these individual blocks? Why does it look different between different species? What actually do these components consist of? In a sense, right, when you get to do the first discovery like that, whether it's a single cell discovery or otherwise, you can ask all kinds of new questions, and your world is opened up in a way that you couldn't have imagined before that particular discovery. So next slide. And actually, there's another sort of conundrum with a very similar origin, which is just like the discovery of viruses themselves and understanding what viruses are. Because before the electron microscope, people knew that people got sick, but they weren't sure exactly what was making them sick. Was it just some kind of liquid, some kind of chemical, something that you couldn't visualize? Or were viruses that you could isolate, right? Particulate. What was their actual composition? And the electron microscope revealed, right, through increasing innovations and with even finer resolution, that the viruses were single particles, as opposed to just having liquid constituents or components, or being really small things. So that opens up again all kinds of other questions related to structure, function, mechanism, understanding. Next slide. And here are just some of the basic questions that I've listed for understanding, right? When you are sitting down and you look at these structures and you're the first one ever to see them, right? What might be going through your mind? Next slide. And I know the QR code doesn't work, but you have this link in your email, and I want to take 2-3 minutes for everyone to answer this question in the form. I think it's a rather important one, which is that, right, you might have your research question, you might be interested in or have done a bulk Illumina experiment or something like that. But I want you to sort of take all of that bias that you have, perhaps from your previous research experience, put it to the side. Imagine that you're sort of looking at this through fresh eyes, and ask yourself, what kinds of questions could I ask about single molecules in terms of the biological context and questions that I'm interested in? So if I'm a virologist, and I could look at single molecules, what kinds of questions would I want to interrogate about them? Versus, oh, maybe you're a plant biologist, maybe you have some other biological topic, but reframe it, reimagine it, and submit your answers to the Google form in 2 minutes time.
And so to close out for the last kind of 10, 15 minutes here for the day, we're going to try and keep all the lectures for the course to be, 3:30, 5:30. I know it's the end of the day. People may have certain things to do in the evening, but we'll and sometimes we'll guest speakers. We want to be respectful of time and try and keep things in that tight window. And then the lab work will sometimes go a little longer. It just depends on how things go. Of course, in the lab that can be a bit more variable. But to close in the next sort of 15 minutes, there was a really great review paper. What in 2008 says, what can you use sequencing for? And the answer is basically everything. You could look at population scale sequencing, genetic mutations. You could screen compound libraries, look at gene expression discovery, and we'll talk a lot about that in 2 lectures. You can look at metagenomics, or complex mixtures of environmental samples. You can look at protein DNA interactions and temporal changes. Basically anything that involves DNA or RNA, you could use sequencing.
Well, I think what's going to have to happen.
Protein.
Industry.
1 second. Oh, there you go. there's also, you'll hear about later, you can actually adapt an adapter to an antibody that binds to a protein. So you can actually look at proteins with DNA sequencers, which sounds counterintuitive. But as you'll learn about shortly, that there's very easy ways to actually use barcodes and then sequence them and see what is something bound to. So, and then here you can see on the left here is what human genome sequencing was, originally, and then this is from around 2016, where it has all these short fragments. But what's exciting is we're now getting to the point where this is getting longer. So this used to be state of the art, but now the single molecule sequencing is long enough that it is becoming state of the art, and it's opening up a lot of new applications and large work. You can see it's over here, this populations scale sequencing. So one of the examples of this is the genomic thing is a 100,000 genomes project, and also the UK Biobank, which is now, it was most of the way through by 2018, is now completed. And eventually they're expanding this to go to 600,000 genomes, and eventually we'll probably get to 1 million. And there's also the United States All of Us program doing this as well. I'll talk about that in some of the later lectures. But you can scale up to deliver results at hundreds of thousands or millions of genomes, and then link that to medical data. There's also a really interesting trial to see if we could actually discover cancer before it's too late. So one of the large studies, what's called the Galleri trial, which is by Grail. There's basically 140,000 people who have no sign of cancer and for 3 prior years. But the question is, what if we could detect cancer just a little bit earlier? This is using cell-free DNA from plasma. And one of the big questions was, well, can you, and how well can you, and how does this compare to standard of care? So this is in the middle of sequencing as we speak, and it is really one of the biggest. There's a lot of, actually, speaking of the Landau lab, there's a lot of work on cell-free DNA and methods to detect small, subtle variations that indicate current cancer or risk of cancer or risk of relapse of cancer. And this is a really exciting study because we'll finally be able to sequence deep enough and enough people to adjudicate how well you can actually characterize early detection of cancer. Because if you want to detect cancer at stage 1 cancer, we want to do it stage 3 or 4, where it's basically too late. You want to catch the cancer right as it's emerging, where you barely have enough cells to pick it up, but you still could pick it up. Would be the hope. So the other thing that's exciting about this is we start to sequence more and more genomes. We start to learn about what are interesting phenotypes that come from a specific genotype, some gene. So, for example, myostatin, if you're homozygous mutation null, you look like this as a kid. If you have it as a dog, you look like this, which is kind of amazing. I think a lot of you might want that phenotype, but you can see they got so big giant muscles. You can also have really strong bones that are almost unbreakable if you have mutations in something called LRP5. And you can also, essentially have something where you can be almost immune to almost all forms of HIV with a mutation in CCR5. And I'll talk about this more in the later lecture, some of these really interesting single mutations that lead to really dramatic differences. So this is, opened up questions here on Earth for, ideas of applications here. But also just last year, we did a survey of what are different alleles you can imagine in spaceflight. If you're going to another planet, what are some genes that you would want for stronger bones, stronger muscles? You want less sleep, controlling aging. So this is still a lot of it hypothetical, but it really opens the door to new ways to look at what the genes are and what they're doing in your body. And then, this is more on the sequencing of DNA, but we will look a lot, as you heard earlier, we're going to look a lot at RNA. And I think this is probably, it's a review for almost everyone here. This is, of course, we have DNA, we have all these exons that get spliced into different components. But it had been historically very difficult to look at us, because remember, all those short read sequencers, you can map them like snow and find spots. But you would have to hope that you would land at a splice junction if you want to infer splicing, these short reads. But the long read is you'll do, and of course, you get the full molecule. You could look at the entire transcript, all isoforms. It gives, this is something that I've been looking at RNA for 20 years, and this is something we've fantasized about really since you learned about RNA, is we want to be able to see the full molecule and all of its variations. And this is just at the RNA and the DNA level. And so one other, I think really interesting fact about this is, if you think about how complex RNA can be, it can be extremely complex. So if you have one exon, you can make one thing. If you have 2 exons, you can put them together and they can either be combined or separate, and you have basically one splice junction and 3 products. You can make 3 things if you splice them together. If you have 3 exons, of course, you can have them individual, or you can make the combinations. You can make 7 variations of molecules once you start splicing them together. And this, of course, continues on and on. If you have 4 exons, you can make 15 variations, and there's 6 splice junctions to keep track of. And this increases at a continual rate of n times n minus 1 over 2, or if you look at all the possible variants there are, it's 2 to the n minus 1 of all possible splicing combinations, which actually means, by the combinatorics, there's more theoretical transcript combinations in your body or in one cell than there are atoms in the universe. So this actually means you, of course, can't make all those transcripts because there's literally not enough atoms in the universe to make them all, but just the complexity of what's possible just in any one of your cells, so it's that vast. But historically, it's been hard to study them all, even just to get a sense of this, because we're using short reads that might pick up here, maybe just the split structure would map correctly, but now we can see it a lot more clearly with a lot of the long read sequencing methods. And so that's a little bit exciting about RNA, and we're still discovering RNA types and genes themselves. So this is from one of my first books, just looking at earlier figure in the book of how many genes did we have in the year 2010. You can see we had about 50,000 genes, and by 2020 we had about 63,000 genes. Now, does that mean that genes were hiding somewhere? It just means they were either expressed, no was the answer, but it means they were expressed at low levels, or we hadn't yet discovered them. Or you can see what's interesting is back, 30 years ago, we thought maybe there were 100,000 genes. And the number of protein coding genes has stayed about the same this entire time. It's about 20, when years once there's 20,000 genes, there is there's 20,000 protein coding genes that actually make proteins. But if you look at all the other kinds of RNAs that have become small, non-coding RNAs, link RNAs are long non-coding RNAs, there's also pseudogenes in the body, and I'll talk about this more in later lectures. There's all these other kinds of RNAs that are active in your cells that have their own biology that control which chromosomes are activated or control some instruments that link RNA hot air. So they have this, real complex biology behind them. And this, doing more sequencing has also helped us find, do deeper sequencing of RNA, you find new genes, new transcripts. You do deeper sequencing of vectors.
Naive question. For the number of protein coding sequences, does that, when people say 20K, do they mean with isoforms or without isoforms?
Without, they just mean the number of loci that makes a protein. If you include proteoforms or different isoforms of proteins, that gets in the hundreds of thousands or millions. It looks very much like this. If you did every possible combination of amino acids that are alternatively spliced and then turned into a peptide, it would also be very vast. It would be the number of possible combinations, which actually I should make a slide that shows the protein landscape as the RNA. So yeah, it basically means the loci, for example, hemoglobin, there's an adult hemoglobin, alpha and beta, and there's two loci, but there's splice, because as evidence here, there's many splicing variations, which then each one of these would then lead to a slightly different protein. So there's they're I'd say very close to each other. A number of possible protein isoforms and transcript isoforms are very similar, and they're both fast. But the number of actual loci is really narrowed down to just those 20,000. And this is from GenCode Genes, the website that annotates it. But so if you sequence deeper RNA, you'll find more functions, more rare transcripts. If you sequence more DNA, you can even find new species. This is from a review paper that showed how many projected new species will keep being discovered over the next few decades. It's expected that we'll keep finding new ones if we look into rare environments. And then also, if you think about the central dogma, if you remember how it started, really it's just, we'll talk about this in the course, DNA, RNA, protein, something about biology. But given everything you just heard and everything to learn about, if you think about it, it's not just that simple. There's many kinds of non-coding RNAs that are guiding us and mediating. There's transfer RNAs, there's ribosomal RNAs, there's other components that lead to proteins. There's also DNA transposons, not as many in humans, but in other species that can copy themselves. And there's also information can flow backwards. You can have reverse transcription, or RNA binding proteins can actually modify the RNA, so you can actually have this information flow backwards. There's actually, DNA copying itself, it seems weird, and prions kind of freak me out. People think about prions and metabolisms, it seems creepy to have a protein touch another protein and then it copies and changes it like that. Like it's just a weird villain in a movie. And prions are freaky that way, but ribozymes can copy themselves as well, and there's DNA transposons. So actually the ability to self-replicate as a molecule, something as across multiple layers of the central dogma. And of course, you'll learn a lot about the epigenome in the course. You'll also get a deep dive in the epitranscriptome, which is all the landscape of RNA modifications. And if you think about post-translational modifications, think about how proteins are modified, you could call it the epiproteome or post-translational modification. Nobody calls it the epiproteome. Again, in PubMed, you'll find out maybe 6 by now. But I think for prefix consistency, I published this a while ago and said if we're really consistent, we call it that. But that did not win the culture battle of molecular biology, so we just call it post-translational modifications. But I still have to point out that if we were consistent, it would look like this. But in any case, there's also other things to learn about. You can inherit modifications of epigenetics potentially through generations. Definitely in mouse models, very likely in human stressful environments, or maybe some intergenerational impacts of stress that's inherited. There are viral-induced RNAs that can be passed on through multiple generations of C. elegans called viRNAs, or you can hand an RNA molecule down. There's also something called maternally deposited transcripts that are in eggs. It's the first cell of an embryo. And prions can through moving between species and generations, and so freak me out a little bit, but there they are. So this is what I like to describe as the more expanded central dogma of molecular biology, and what you can think about. And that does extend this red part here. These many millions of permutations of RNA modifications. So we now know of basically at least 100 types of DNA modifications. Now approaching actually almost several hundred epitranscriptome modifications, and many just classes of post-translational modifications. Again, the raw numbers, you go back all the way here, the raw numbers are vast. There are many, many more, but if you just actually look at, there are, the types of them are hundreds of types of modifications. So if you look at one locus in the genome and how the number of ways it could be modified, it is large and complex. But we now have many tools that let you map them and see them. We're learn from one over the next semester. So that's it for today. So this is a quick summary of the first lectures that I think I spent a lot of time in microarrays, and I'm very glad that no one here had to spend time on microarrays, but it was a much slow and laborious way to understand biology and splicing and function. But today, you can sequence anything anywhere in Earth and in space, and actually allows population scale cycling, population scale questions to be asked. A lot of work that you can do with single molecule resolution was unimaginable 10 years ago, and we can really change what kind of questions you can ask. And we'll talk that again some of the homework, and you also learn about that over the next few lectures. And as Theo was mentioning, in discovery of just cells and viruses, just finding the fundamental unit of the biology, finding the cell, the unit of the cell or the molecule, really opens up the brain, to the field, to new ideas and new directions, which is very exciting. And what is really happening now with sequencing. But you always want to have standards. You want to have sanity. That's why I pointed them out. And it's one of those things where it seems really boring to have to do a standard and run a control, but until you can run the control correctly, you can't talk about that weird sample that you got from space, or a really unusual mutation you found in a tumor sample. Something that seems really unusual. You have to make sure you did everything else right to make sure you can actually describe it and classify it. And it's also, again, this is just another versions of lectures. And finally, the exit ticket in the last few minutes. We want to hit this. It should be in the link in the email. Again, let me know if you don't have it. But this is the last exit ticket for the first lecture. And again, this will be every Tuesday at 3:30 to 5:30, and there'll be some other lab sections. And that'll be happening in our lab later this semester. And then we'll be a little bit of time for any questions. I have a few exit tickets to